Now, so everybody, thank you for coming today. My name is Mrksich. I'm the president of US HUPO. And today we're having a webinar that we call Best of US HUPO. And this is the best of all the talks, but there were so many talks, so it was very difficult to choose. But we tried to pick the very impressive work that was presented at our annual conference of US HUPO, which was in March in Chicago. And, I'm really happy that everybody is here today, and this will be recorded and we can distribute this, and it will later be posted on our website. So I'm really happy to share this with my co-chair, Jeff Savas. And Jeff, I'd like to turn over to you to briefly introduce Milan, our first speaker.
Good morning, everyone. Thanks for joining us. It's my honor to introduce today's first speaker, Milan Mrksich. Milan is the VP of Research currently at Northwestern University and also serves as the Henry Wade Rodger Professor of Chemistry. Milan has a very distinguished academic career. He performed his undergraduate studies at the University of Illinois, Champaign-Urbana, downstate. He did his PhD at Caltech and a heavy-hitting postdoc at Harvard with George Whitesides, one of the most distinguished chemists in the world. He started his independent career at University of Chicago and then migrated to Northwestern, just a bit north up the lake soon thereafter. Milan has published more than 240 papers with an H-index greater than 98. And his interests really lay at the interface between chemistry and the synthesis of surfaces that allow us to understand biological environments. With no further ado, Milan, take it away. Thank you.
Well, thank you, Jeff and Birgit. It's good to be back for the online session that we're having today. So I'm going to share a screen.
Oh, sorry, Milan, no, you're muted. I'm sorry.
Now I should be there. Okay.
Yes, sir.
Thanks very much. Sorry about that. So in the half hour we've got, I wanted to give a description of a story that started about 20 years ago in the lab that really led
to interrupt one more time. I don't think we see the slides yet.
Oh, I just shared. Let me try again. How about this? Can you see the slides?
Yes.
Okay, perfect. All right, now we're all set. So what I wanted to do was share a story of the development and ultimate implementation of a label-free high-throughput assay, a biochemical assay for enzyme activity that we call SAMD. And let me kind of just use a slide to start at the end, which was a few months ago. We sold a company that ultimately we started from this technology, sold it to Charles River Laboratories. And this is a company that is the world leader in label-free high-throughput assays for screening, for high-throughput screening in the pharmaceutical industry. And you can see a little bit of a press release there from January of this year. And let me go back because I think it's interesting the way that this initially got discovered. The different things that we did to develop it into a robust quantitative high throughput assay. And then the way that the company got started and got going, which was a non-traditional model as well. So this story really started, as I said, a bit over 20 years ago. At the time, we were using the self-assembled monolayers, and these, of course, are now well established as a really well-defined and controllable surface chemistry with alkane thiolates chemisorbed to a gold substrate. And what you see here is an example, our first example of performing a kinase enzyme assay. And so the monolayer has chains that terminate in a benzoquinone group. We use that for conjugation or immobilization. And then also these ethylene glycol oligomers, which make the surface inert or resistant to nonspecific protein adsorption, which is critical if you're trying to control interactions at the interface. And so the peptide here is functionalized with a cyclopentadiene. From organic chemistry, people might remember the Diels-Alder reaction. It's a really good reaction, similar to the click reaction that Carolyn Bertozzi and Barry Sharpless developed. And this gives us immobilization. And in this system, the peptide, if the surface is treated with a kinase and ATP, leads to phosphorylation of the tyrosine residue. And the way we would characterize that is to either use a radio-labeled phosphate, that is ATP source, and then look for how much P32 is on the surface. And you can see in the first bar, there's some amount of it. These other experiments are controls of different sorts, including the last one where there's no P32 added. So that's really the 0 point, the background. And what we were excited about was it was a very clean assay, signal to noise of 100 to 1. We showed that it was quantitative and that we could measure biochemical constants. And we were excited that this could be a high throughput format. The challenge for us was that you still had to label the reaction product if you apply this to an enzyme activity. With kinases, that was P32. With other enzymes, it could be other strategies. And many enzymes, it's extremely difficult to work in a label to do assays. And that's when, as you see on this slide, we tried lots of other label-free methods, including the surface plasmon resonance methods, other optical methods. But we found that MALDI-TOF mass spectrometry was magical in this context, because if you put a monolayer into the MALDI instrument, when you hit the sample with the laser, with the pulse, the monolayer desorbs from the gold substrate. The thiolate gold bond cleaves, but the alkane thiols pretty much stay intact, very little fragmentation. And so you get very clean spectra that reveal the masses of the peptide alkane thiol conjugates. And so in this example, again, a kinase activity, we're immobilizing a peptide, this time using a thiol from a cysteine residue reacting with the maleimide group on the monolayer. And when we immobilize the peptide on the upper right, you can see the spectrum, one primary peak at a mass of about 2100 Dalton, which corresponds to the expected mass. If we then treat that surface with Src kinase and ATP, rinse it off, take another spectrum, we see that the mass is shifted by 80, which is consistent with a phosphorylation reaction. Now, I should also say that when we do these reactions, we do add matrix, and we know the matrix is important for minimizing fragmentation of the ions. And so they are energetic when they're desorbed, and they can fragment if the matrix isn't there to pull energy out. But it was extremely exciting because now we're seeing mass of the substrate and product. We're able to do that in a very simple way, and we were immediately excited about the generality of the technique. And so here is another example, the deacetylase assay. In this case, we make a peptide where the lysine side chain is acetylated. We immobilize it. You can see the spectrum. You see a few peaks. These are the revealing both isotopic abundance of the molecule with a little bit of C13 in there and some proton adducts. But what's clear is when you treat this now with a deacetylase, KDAC8 here, we get deacetylation. You see in the spectrum, the peaks shift lower by 42 Dalton, again, consistent with the loss of the acetyl group, and very clean. So we did this for a number of enzymes, lots of different examples over the years, and just summarize it here. The technique, and we called it SAMD because this is self-assembled monolayers, or SAMs, for MALDI. So when you put those together, we refer to it as SAMD. And it's exciting because it's applicable to just about every biochemical assay. All molecules have a mass. It's only the isomerases or racemases that don't have a product with a different mass that this isn't obviously applicable to, but it's extremely general. we see both substrate and product. Most labeled assays, you only observe the product. And so we've shown that that lets us be more quantitative in terms of yield. Because we're not using labels, we're able to observe products that were unanticipated, which means we can discover reactions that were unknown, something that's not possible with the labeled assay. And then this last part, I'll just skip to high throughput formats. It turns out that these are very adaptable to the standard 384 and 1536 well formats. Those are plates on the upper left that are in that size, stainless steel with an array of gold islands that are deposited by evaporation. You can see that they are the exact geometrical formats, and that means that they're compatible with all of the liquid automation and robotic tools that are used in the industry. And, I don't know if the movie's going to come across well, maybe not. So in terms of the mass spectrometry, we use both the now the Bruker instruments, the really fast MALDI Rapiflex instruments, but also the SciEx instruments, the 5800. And we can scan about an assay a second. And so everything is now automated. Everything is kind of analyzed through a lot of different root scripts we have to look at the data that comes off. And we can run really now a couple 100,000 experiments in a day between 2 or 3 instruments. And so really exciting. So what I want to do next is show you a few examples. I know we don't have a lot of time of how we've used this in experiments, what it's enabled. And then I want to say a little bit about how we started a commercial effort based on this. So the first example, and that is identifying or developing reactions, typically organic reactions that can modify peptide sequences. And so this is important for conjugation of drugs to proteins and antibodies, important for labeling them with other functionalities. And one of the key things is not just developing a reaction that labels a given sequence of peptide, but doing it selectively or specifically so that it's controlled and it's not reacting in other places. And we recognize that if we had an array of peptides, hundreds or thousands of different sequences, we could treat that array with a set of reagents and then scan it with mass spec to see where the reaction products were and where they weren't. And so we're not just selecting for peptides that react. We want to know quantitatively where the reaction's happening, where it's not, so we can understand the specificity. And the first example was really meant to be a control experiment. It turned out to be interesting. What you can see is we created a library of peptides where 2 amino acids were varied between all possible dyads. We treated that with acetic anhydride, expecting it would label lysines because of the amino groups. And you can see in the spectra on the middle one, here's a peptide that when it was treated, we shifted from the unacetylated form to the acetylated form. So this heat map on the right is just a representation of the percent yield of acetylation for each peptide, where we show the amino acid residue in the X position, and then on the top in the Z position. The first thing you see is wherever we have a lysine, the K is here or horizontally, there's some acetylation. That's expected. It's not great because those lysines are protonated, so it's only the small fraction that's not protonated that is able to react. But then you see 2, where again in grayscale, black is 100% reaction. 2 of these sequences gave us complete reaction. When we had a histidine after a tyrosine, or a tyrosine after a histidine. So that histidine tyrosine dyad gave us very good reactivity. And we were surprised that this wasn't known. In terms of the chemistry, it makes pretty good sense because histidines with the imidazole group are very nucleophilic and they will acylate, but they're also easily hydrolyzed by water and go back to the substrate. But if there's a neighboring tyrosine, you then get transfer of the acyl group to the tyrosine, and a stable ester is formed. The tyrosines on their own are not sufficiently nucleophilic to do that. So we could confirm that that's in fact what was going on. We discovered a new reaction that's specific for these HY dyads. And it really reveals the power of being able to see all of the reaction products and being able to look at lots of substrates to really confirm specificity, something that's been challenging to date. Okay, let me give another example. This is one where we were interested in a longstanding program with Mike Jewett's lab of how do we get efficient glycosylation of proteins where we want it, and to be able to elaborate those carbohydrates into more complex structures. Our approach was to develop peptide sequences that were extremely good substrates for a given glycosyl transferase, the enzymes that attach a sugar to a amino acid residue. And then we could stitch that sequence into a protein. And then if we treat the protein with the glycosyl transferase, we should get specific and efficient glycosylation. So what we've done here is we're using glycosyl transferases that glycosylate at asparagine, this N residue. And we wanted to vary the amino acids before and after, knowing that the +2 residue should be serine or threonine. Now, this was neat because it was done in a high throughput format. We had 384 well plates. Every plate had the enzyme machinery, which was produced with cell-free protein synthesis, had the peptide substrate, a different sequence in every well, had the sugar donor, and the enzyme. So we ran the reactions, then we stopped them. We, with the robot, transferred each of those reaction mixtures to a monolayer array plate, as I showed earlier. The peptides underwent immobilization because they had a cysteine residue that reacted with the maleimide. And we immobilized both the substrate peptide in black and the product peptide in blue. And then when you take a mass spectrum, you can see both the substrate and at higher mass, the glycosylated peptide, and you can calculate the yield. And that lets you then plot out, again, in purple scale, since we're at Northwestern, plot out the degree, the yield for glycosylation. And so on the left, you see again, the -1 residue vertically, the +1 residue horizontally. And you can see there are sequences that are very efficiently glycosylated. Most are not, that's expected. And I'm just going to point to this one outlier, which is a very good substrate. And this is odd because it's a tryptophan, a very large amino acid, next to a glycine. Tryptophan next to alanine up near the top gives some activity, but then the activity falls off. So tryptophan next to a small residue seems to be the trick. That wasn't known. These assays are typically done with HPLC, very challenging. And the ability to look at lots of peptide substrates, because it's now a simple assay, let us identify these new specificities. So I'm not going to kind of belabor what we did next. We basically kept expanding our library where we would fix the -1 and +1 positions and start to then vary other positions. And we could find better substrates as we built out optimal sequence, and we could keep walking out. We probably looked at a few thousand sequences in this paper, and we ultimately developed sequences that were far better as substrates for the enzyme than what was known in the literature. And the best one did have that tryptophan glycine motif, something that just hadn't been observed. We also show that if we stitch those peptide sequences into a protein that we express and then treat the protein with the glycosyl transferase, we get efficient glycosylation, and the relative activities of the peptide substrates lined up with the activities of the protein substrates that were stitched with those peptides. So really an exciting capability to radically accelerate the ability to come up with good substrates. And I just want to draw the distinction. You could do screening in a combinatorial library, have lots of peptides, treat those with an enzyme, fish out the products and decode or sequence to know which sequences were good. But we need more information. We don't just want to find winners. We want to find substrates that are specific. And so we need to characterize a large sequence space to know that a given enzyme, if we're looking at several enzymes, what its specificity is relative to the others. So you have to see the winners and the losers to do that. And so these aren't high throughput combinatorial libraries. It really is high throughput experiments where we're getting quantitative data on every reaction that we run. Okay, I am gonna, again, because of time, I'm going to jump here and talk about phosphatase assays. Those of you who measure phosphatase activities know that these are challenging assays. There's a malachite green assay, which measures the phosphate ion that's liberated by a phosphatase. Those are clearly hard to do with cell lysates because there's a lot of phosphate in the background. And so you're looking at a change in phosphate level relative to a high background. But again, with SAMD, it's really straightforward. We're just looking for loss of 80 Dalton from a phosphorylated peptide. So just like the arrays earlier, what we were able to do was create arrays of hundreds of peptides. You can see here on the upper right, it's a phosphorylated threonine with the amino acid before and after varied among all possibilities. And so that array we use by applying a cell lysate and then analyzing to look at a fingerprint of the phosphatase activity in the lysate. Now, we're not looking at which phosphatases in the lysate are responsible for which reactions. We're really using this to look at the global profile or activity of phosphatases, which is a function of which phosphatases are present. So what you're looking here is a spectra on top. We've looked at those. But on the bottom is a heat map of phosphatase activities, again in grayscale. Black is full dephosphorylation. And I'm just going to come here. We did this with mesenchymal stem cells that we cultured and either induced an adipogenic differentiation or in the next slide, an osteogenic. But here, if we compare on the left, lysate activity profile for the mesenchymal stem cells, and then in the middle, that same cell line after we've induced adipogenesis, we can then take a difference between the two, and that's what you're looking at on the right. So green means the activity is higher for that peptide in the differentiated lysate. Red, it was higher in the non-differentiated lysate. Now, if we also do that for osteogenesis, you see the map on the right, it's different. And that's expected. We expect a different complement of phosphatases will be expressed in the osteoblasts. If we then basically compare what's different between the adipocytes and the osteoblasts, we get this map. And so what we're looking at is, that higher activity at this tryptophan and this tryptophan, higher activity in the osteoblasts really caught our eye, and then lower activity for some of the others. There's a real difference between these two differentiated lines. And what's interesting is that one of the markers that's used for osteogenesis is staining for alkaline phosphatase. And so we said, what's alkaline phosphatase's specificity? We just put the recombinant enzyme on the array, and this is the specificity we found. And what you can see is it prefers these tryptophan residues at the phosphothreonine. And if we compare the enzyme specificity and the difference map in the lysates, you can see that a big part of the difference in the lysates is because of alkaline phosphatase expression. So that's really satisfying that, again, we can look at these lysates, map out phosphatase activities across hundreds of peptide substrates and get data that makes sense, that's reproducible, and it really represents a new capability for characterizing these lysates and looking at the omics in them. Okay, I am now going to say a few words about the company, because this was also an interesting thing for us. We were at the point around 2010 where we really recognized that we had a high throughput assay. It was quantitative, and we were really looking at how we could commercialize this. And ultimately, it was commercialized in the company called SAMD Tech. But just to tell you how we did it, Mike Scholey on the upper left there was a scientist I worked with a lot. He was in the Bay Area working at biotechs, working with Pim Stemmer, the late great bio entrepreneur in the area. But Mike was looking to come back to Chicago. We talked, he came into my lab, and we had some unrestricted funds that he could use to do these things, develop some prototypes, look for vendors who could make the plates for us, do some financial planning on what a startup in high throughput screening would look like, write grant proposals to get some SBIR funding, non-dilutive funding that would let us get going. And very importantly, we reached out to people in pharma and HTS groups, but the way we got in the door was we looked for alumni from University of Chicago. They all took our call, spent some time with us on a WebEx meeting, and in every case, we're really impressed with what we had and wanted to go further and see if we could do a non-disclosure agreement and learn about their actual programs. Well, that's just when we got an SBIR funded, and we then started a company. Carmichael Roberts on the lower left is a longtime friend, and he and I have started a number of companies together. So he was really he's now a venture capitalist with his own funds, but he had worked with us to get the company set up. And we actually got it set up based on that SBIR grant. We found a first client within a month. This is the company that was running deacetylase targets, and there weren't any good assays. And so we reached out to them, and they gave us a try. They basically paid our bills for the first year without knowing it. And we slowly grew from there over 10 years to the point that we've now worked for 75 companies or so, including most of the global large companies. We do all the standard things in high throughput screening. We, sort out the enzyme, the conditions for good enzyme activity, show that it's reproducible, it works over plates, and then do the high throughput screening. And about 4 or 5 years ago, we entered a partnership with Charles River Laboratories, the global giant in the services space for early stage drug development and discovery. They basically partnered with us so that they could sell our service. We could get access to their compounds. And then earlier last year, about 15 months ago, we expanded the partnership. They actually bought a part of us, about 20% of SAMD Tech, and then integrated our service into their offerings. So their global sales force was selling SAMD as the label-free high throughput screening technology. You can see on the right, 75 companies at any time we're working for. And we then added lots of other formats, not just label-free. And then this is the slide I started with. In January, they bought the other 80% of SAMD Tech, and now it's one of their business lines. And so it's an exciting story because we never raised money from investors. We bootstrapped it early on. And because the technology really hit a sweet spot that was extremely challenging with other approaches, we're able to just grow it and organically add clients and learn a lot about the industry and how these things are done, and become a central player globally. So that's the story. I'm going to stop there. I know there might be time for a question or two, but it just is a really powerful technology because the label-free nature is extremely important, and it is pretty simple in the end. It's quantitative, it's high throughput, and that makes it really useful. So I'm going to stop there, Jeff, and we can take any questions if you like.
Oops. Yeah, thanks so much, Milan. This was a wonderful talk. Okay. Yes, thanks so much. Maybe I can actually start with this. So, for example, if now with this fascinating technology, how can people get involved with such a technology? Would they contact you or the company? And where do you see this going for the field? You know, what is your vision for all of this? You hinted a little bit at it.
It's a good question, Birgit. So the company, Samdi Tech, which is now a business within CRL, serves the pharmaceutical industry for high throughput screening. That is their sweet spot. But as I've tried to show, because I didn't show examples of HTS, this ability to do high throughput experiments is extremely powerful in science and basic research. And we've had many examples where, and we really do focus on experiments that just can't be done in any other way. These include also reaction discovery and organic chemistry, having functional groups on a surface, treating with different reagents, and finding things that are unexpected. Most named reactions in organic chemistry were not intentionally developed. They were a recognition of something unexpected. And that's what this is really good at. A lot of work in metabolomics, where we're looking at biosynthetic pathways, and we're able to capture all of the intermediates and look at every one of the intermediates quantitatively, so we can do pathway profiling and discovery. So wherever there's a need to do a high throughput experiment where you need hundreds of thousands of discrete experiments to kind of draw the answer out, this is powerful. So at the moment, it's not a commercially available system. The plates aren't available commercially. We collaborate with a number of labs to do interesting things. So I'd invite people to send me a note, reach out, and if there was something that, you know, made sense in terms of what we can do and the nature of the problem, we'd be eager to talk about it.
Thank you.
I have one question. What a cool talk, Milan. Thank you. I'm curious if you could tell us a little bit more. So say like you had a test compound or a drug, how would you use your platform to test the efficacy of the drug, or how does this integrate with Big Pharma?
So the core service is that early stage where they validate a target and they then want to develop an inhibitor, agonist of the enzyme target. And so they would run this assay with that enzyme activity, you know, 1 million times, and a different small molecule in each of the 1 million wells to find hits, to find the compounds that block activity. And then once they have the hits, they can figure out which ones to pursue and do med chem around. It's
one assay with many different compounds.
Yes, yeah. Now there's another interesting application, Jeff, that is starting to happen, and that is, let's say you pull out, you know, 40, 50 hits, which would be not unexpected in a large screen. You might look at those and they might fall into 3 structural classes. You can kind of say, okay, here's one scaffold, here's another. And so which one do you go with? So one of the things that we're also interested in is treating cells with these different compounds and looking at the lysates and asking the question, like in that phosphatase example, does the pattern of enzyme activity, is it the same for the different classes or different? Which tells you something about whether they're operating by the same mechanism or whether they have off-target effects in the cell. So looking at the lysates globally gives you a sense of whether compounds are mechanistically similar or might pick up another target? And are they just altering the activities you're looking for to alter, or having a broader effect, which might be bad and steer you away from them?
Makes a lot of sense. Thanks for that very interesting explanation.
That's wonderful. Yeah, thank you so much, Milan, and thanks for taking the time to work with us here. And this was very inspiring.